Uh, is Thank you very much, Nicole, and Rachel for the invitation. I think I can already share my screen, and here we go. I'll put it in presentation mode, and let me know if you can see it. And uh, I also, can you see my cursor when I go by? I can't find the tool for giving the, it's not opening up the tool for giving a highlight, but I can just use this. That's good enough. Uh, so anyway, I wanted to talk today about, I know Chile is very far from you, uh, but I wanted to talk about why a lot of people like to come to Chile to study Chilean waters. They offer a set of natural labs for studying phytoplankton in a changing ocean. Oops, I have to go back to, um, I just did something. I can't see my own screen now. So let me go back here. There we go. There we go. Yes. So I wanted to also just mention some of my funding organizations here, my own university, but also getting funding from the Chilean National Fund for Investigation and Development. And here's a picture of me aboard a boat. And here's a picture of some of the organisms I like to study. Okay, so phytoplankton in a changing ocean. There are a lot of global changes today affecting phytoplankton. Some of them you've probably heard of, a lot of these. Ocean acidification, which is the increase in CO2 dissolving in the ocean, leads to a drop in pH as well as a drop, curiously, in the carbonate concentration. I'll explain a little bit later more about that. There's also a loss of oxygen and potential expansion of oxygen deficient zones as the ocean warms. Part of that is related to another effect which is occurring is the increased stratification that's occurring at low latitudes, and that intensifies the coastal offshore gradients because there's also oftentimes at mid latitudes there will be increased upwelling at the coast for increased winds, at least for a period of several decades. Of course, there's also eutrophication of coastal and estuarine ecosystems, like the Baltic, for example, experiences that famously. So does the Gulf of Mexico. And there's also occurring as transport of species. But Chile is an especially interesting place, or the South Pacific as a whole, right here, is interesting for these phenomena: ocean acidification, the loss of oxygen, and the increased stratification and intensification of gradients. Uh, Here, let's focus on this region. Here's Chile. Here's Peru. Here's I've marked in white are the economic exclusive zones. And here we have along the coast we have a lot of highly productive waters that are also, as well as being high in nutrients, they are naturally high in CO2 and low in pH and oxygen. Those conditions, in fact, chemically, in that sense, they are actually similar to what is predicted for the rest of the ocean after the year 2100. So they already naturally experience that condition. Chile in the south, we have a really a large system of canals and fjords and inner seas with highly varying salinity. For this would be something similar to what in your neighborhood, what is in present in Norway. This is a another very large system of fjords. So one has these um, also chemical changes doing varying salinity with glacial melts. And we have the greatest productivity gradient in the world. From the clearest natural waters are close to Easter Island, right about here, and with these among the most productive waters along here. So that's a very big gradient in productivity and interesting to study. So I like using that as a natural lab. I'm going to start with the oxygen and CO2 issues. So here we have a section. This comes from a paper published by Christian Vargas in 2021. It was a cruise my research center did in 2015, and this is a cruise along the northern part of the Chilean coast and then offshore a bit up here. And here we have above, we have oxygen. When the purple colors is where oxygen goes to very low levels. In fact, the sensors are not sensitive enough to low levels to detect how low the oxygen really is, but zero. We know it's zero because they also accumulate nitrite from what happens at low oxygen, at when you have anoxic conditions. Further to the south, you have low oxygen but not anoxic conditions. So you actually have quite this gradient. Those waters also, here you see the CO2 levels, and the CO2 under these subsurface waters are also naturally very high. So they're naturally at levels uh, 3 times or more of current atmospheric levels. So of course, when there's upwelling in these waters, you have natural exposition of the surface to low CO2 and low oxygen, or low oxygen and high CO2 conditions. Well, one thing that I was quite startled to see a few years ago, this was known about, but I was really startled to see about it, and nobody had an explanation, is that in these waters in front of the north of Chile, above the anoxic conditions, you sometimes will get, here you have the oxycline, here's the profile of oxygen, and if we look at the profiles of fluorescence and transmittance, you'll see 2 peaks instead of just one. And there'll be one peak above of fluorescence above the oxycline, and then another one below. Below, there's enough light to develop a community of phytoplankton, and that community of phytoplankton turns out to be dominated only by, and not by, the typical deep phytoplankton you find in the rest of the ocean, which is normally diatoms and coccolithophores and dominate it. That one is populated only by 3 specific lineages of Prochlorococcus that are only found there, that are anoxic, specific to anoxic waters, and not the typical deep Prochlorococcus you find in the rest of the world. The primary chlorophyll maximum is composed of diverse phytoplankton like in the rest of the world, including eukaryotes and cyanobacteria like Synechococcus and Prochlorococcus. And so in between here, you have not only a chlorophyll minimum, but a transparency maximum. And in fact, even when the secondary chlorophyll maximum doesn't develop, one typically in the oxycline has a transparency maximum because a lot of cells simply disappear there. Well, we've been studying a lot. I was recruited by Osvaldo Ulloa to collaborate with him and his students studying this secondary peak. He was really interested in these anoxic Prochlorococcus, but I got more and more interested in this. Why is there things disappearing there? And we were just about to start experimental studies on it. And we're just starting those when the pandemic hit and we had to do it from our office. And so this is a paper published led by Jane Wong, who we published in Limnology and Oceanography last year, where we're looking at this and we're trying to review what it could possibly be that could be causing this. And we proposed one possible idea is to do a very simple model of what happens if you consider just light and nutrients. Well, if you consider just light and nutrients, it's we think a simple photosynthetic model, you should get a curve that looks like this in orange. But if we assume that the lower peak is of organisms that are inhibited by oxygen, and the upper peak is organisms that require oxygen with some reasonable parameter guesses, in reasonable range of parameter guesses, we would get often this. That would be something very similar to what actually happens is you get a missing photosynthesis, like there's a missing phytoplankton there. So one possibility we're discussing with John Raven, who was a famous phytoplankton photosynthetic physiologist who unfortunately died a few months ago. But we were discussing one possibility is could photorespiration or water cycles be required for most modern phytoplankton? Or another possibility that we didn't review there, but we're also considering, is there something that's inhibiting them from growing there? Here's a paper on using the Prochlorococcus that are found in most of the ocean. That in the lab, this is by Bagby and Chisholm in 2015, and in their experiments, one can see that very low oxygen concentrations do inhibit the Prochlorococcus that are found in the rest of the world. And so they are inhibited by low oxygen. This is a paper we published. We didn't get to complete our experiments because the pandemic hit, but we did at least get one experiment where we tried using high oxygen, high pH conditions and low oxygen, low pH conditions designed to mimic the waters right there. And this is a flagellate Emantonia that was isolated from these waters, and it grew well. in the normal control conditions and just didn't grow at all in the conditions designed to mimic this area. So we're continuing to investigate that. The results we're getting, I'm not going to show now. There's some of this is one of the cruises we went on in April and May, where we were looking at that and trying to test some of these ideas. But we don't have anything really consolidated to show you. But I will say the results are a little bit confusing. So it's not sure what's happening, but why the phytoplankton are disappearing from that zone. So stay tuned. Okay, so now I'm going to talk about something I've been working on for longer, on and off for longer. And I'm not the only one. There are a lot of people who've dedicated their careers to this, which is the idea that ocean acidification affects calcifying organisms. Affects all organisms, but it particularly affects calcifying organisms. Here's a graph of the predicted pH change in the next this century with different possible outcomes for human behavior. And if we translate that into changes in the carbonate chemistry, here we have a drop in the change in pH as CO2 rises. And curiously, the change in pH as CO2 rises, while that it does cause an increase in the carbon dissolved inorganic carbon in the ocean, it actually leads to a drop in the carbonate ion composition. And that's because the changes the equilibrium between carbonate and bicarbonate. And the calcification reaction net, because most of the carbon dioxide dissolved in the ocean ends up as bicarbonate, not carbonate, the net reaction for calcification is really one can consider with bicarbonate. And in fact, one can consider it this way. One way to consider it is that bicarbonate is precipitating with calcium, releasing. So some of the bicarbonate actually is converted into calcium carbonate, and some is released as CO2. So if you increase CO2, you actually will force this equilibrium to the left, as shown here. So that's one way to think why calcifying organisms would be affected. Well, that's the way of thinking that people started with when understanding how calcification should be affected, but it becomes a little bit more complicated with coccolithophores, as we'll see. And one of the reason is that they do calcification inside the cell. And so that's not so clear how this is going to affect. And it's also not so clear because for calcite that coccolithophores produce, this equilibrium, even if you add more CO2, the prediction is it never will get towards thermodynamically favorable for dissolving the calcium carbonate from calcite. It will for other forms of calcite. So other organisms that produce, for example, aragonite instead of calcite will sometimes dissolve in the upper ocean. But coccolithophores shouldn't have a problem. Yet people have been wondering about this for a while because the evidence in the labs was contradictory and sometimes suggested they would have a problem. This is an example from a paper by Luc Beaufort in 2011. So he said, okay, the lab experiments are confusing. Let's look at nature. And so they looked at living and fossil coccolithophores over the entire ocean and in paleo environments. And normally they found the coccolith mass of one genus, the most common genera of coccolithophores, tended to decline as carbonate ion declined. So as you get higher CO2 and lower pH, you decline. But there was this curious exception, which was found in 2 samples in front of Chile, where you had this very high CO2 upwelling, where instead of becoming thin and degraded, the coccoliths look like this. I don't know if you can, hopefully you can see there that this is the same species as here, but when they're in front of the coast of Chile in this high CO2, low pH environment, they were producing these big, thick, solid coccoliths, which was opposite of what you would have thought. But that was a paper that I was involved in a couple years before had found that there was one strain of this coccolithophore species that actually calcified more under high CO2. And so based upon that, Luc Beaufort said, maybe this is a strain, this is populated by strains that are naturally adapted to ocean acidification. I was curious to investigate that because this actually contradicts pretty much what we know about calcification in coccolithophores at the moment. So this is a summary I wrote to summarize a paper, the information that was some discoveries by Kottmeyer et al. In 2022, and I tried to do this visual summary. So on the outside, we have the equilibrium for calcium carbonate and for CO2 dissolution up here, but the coccolithophore is actually precipitating the calcium carbonate into the coccoliths inside the cell and then exporting them. That's occurring in this coccolith deposition vesicles. So they have to import bicarbonate. Some of that they send to the chloroplast for photosynthesis, and some of it they send into a coccolith vesicle precursor. Now, inside, when you precipitate calcium carbonate, the net reaction to really understand it should be written this way, because really if you are getting rid of using bicarbonate to precipitate it, you're generating protons. And those protons have to be exported. So there's many different ways of writing this equation because there's actually a network of many different chemical equilibriums that are related. So one of the things that you produce is protons, and you have to get rid of those protons to maintain cellular homeostasis. And so it was a puzzle for a long time about how coccolithophores did it, because they would have a huge proton flux. In fact, one can calculate from the amount of calcium carbonate they're producing that they're one of the organisms with the highest proton fluxes that we know about. And so they have to push these protons out. And Kottmeyer pointed out that what they're doing is they're using voltage-gated proton channels to passively export the protons when you get these fluxes of the protons into the environment of the stable environment of the rest of the ocean. If the rest of the ocean is stable and at a pH 8, this works great. You flood the protons out and with a relatively metabolically cheap way to do it. But if the pH drops to around 7.8 or 7.6, then this starts to get harder, and the protons don't go out as easily, and you get degraded, malformed coccoliths. So higher calcification would seem to exacerbate the problem, not help it. So how are we having these high calcified forms in high CO2, low pH waters? So first of all, this is the coccolithophore we're going to be working on today. It's Emiliana uxlei. It's a strange organism, but very interesting because it has exceptionally wide ecological distribution. It forms these large blooms that are visible in these satellite images that are the ocean reflectance converted into calcium carbonate concentration in the surface ocean. And it forms these large blooms at high latitudes and in places like the North Atlantic, the North Sea, the polar front. But it's also present at low latitudes. Even if it doesn't show up on the satellite images, it's always there. It's also curious because it's only appeared in the fossil record 291,000 years ago. So that's about the time scale of the separation from humans from Neanderthals to give it. So in the time scale that humans colonized the surface of the Earth, the terrestrial part, this organism colonized the plankton all over the ocean. So that makes it interesting. And another thing about it is that with flow cytometry, I can distinguish it based upon the fact that these are coccoliths here around this light image, and they depolarize scattered light. So if I look at depolarized phytoplankton that scatter depolarized light, here's light that's polarization perpendicular to the laser and light that's in the same polarization of the laser. These ones up here, they're all, sort them out, they're all coccolithophores in this genus. And so I can collect them, and even when they're at low concentration. And so we've been studying them along in the South Pacific, both looking at them and just taking filters and looking at what's naturally present, and then getting them in the lab to study them and studying different things about them. But one of the things that was really interesting to study is are they really this high calcified form present in these low pH waters? And here we have the pH that I sampled, and here we have what's there. And this data, this is what published in 2018. We now have even more, probably 10 times as much data showing exactly the same thing. This form consistently is one of the dominant forms present, the phenotype with these that's over calcified, where it's the overgrowing the calcite is present in these low pH waters. So is it naturally adapted? So we get it in the lab, and we put it in, and we compare it to strains that have a more normal phenotype from neighboring waters. And here we have uh, look at, and we expose them to the conditions that are found in these upwelled waters in terms of CO2 or a control condition. And here we have the growth rate. And in both cases, the growth rate is lower when you have high CO2. So both of them are inhibited by high CO2, and there's no difference. If we look at production of organic carbon, no difference. If we look at the calcite production versus organic carbon, no difference. I don't show it here, but we also looked at malformation of coccoliths, no difference. This high calcified form isn't any specially adapted to these conditions, at least in the laboratory. But if we look ecologically, we can do, and here's where kind of more data, and we keep getting more and more data that keeps saying this. Ecologically, this form, we call it the R or hypercalcified form, that this one, this is an outlining mean index analysis, and ecologically, we compare it to the conditions it ecologically likes. It likes uh, low pH and high CO2. So it seems it has a wide tolerance for this. But in general, the species Amelia uxia is a very generalist with respect to chemical conditions compared to other closely related coccolithophores. So the ecological occurrences are contradicting what we see in the lab, and we still don't know how to resolve that. But so I've told you two stories now where we don't have complete answers. Okay, so I also started, the reason I started getting this was students here kept thinking that, oh, coccolithophores must be really important in the coast of Chile because Peter's studying them. And I said, actually, no, I'm studying the things that are, because they're actually low abundance here for some reason. And so I thought I'll study something that is actually more clearly important so that and also that it's easier to justify why I study it. And I decided that something that'd be a really nice complement to the coccolithophore work would be Pseudonychia. The coccolithophores, remember I said there's this one species that dominates the whole world? Well, for among the coccolithophores, well, in Pseudonychia is a contrasting example because this is a genus that where there's a lot of species, and the genus is all over the place. It's found in really diverse places. Is a pennate diatom that relatively recently adapted to the planktonic existence from its benthic ancestors, but it's important in both in coastal upwellings and open places, but it's filled with a ton of different species. So instead of having just one species dominating all the coccolithophores, the diatoms are the absolute opposite. They always have tons of species, and Pseudonychia is no exception. And in Pseudonychias, they all look pretty much the same. So functionally, it's really hard to distinguish them because morphologically, it's hard to distinguish them. And we can only do it by molecular analysis. However, one functional difference some of them have is that half of them produce domoic acid, and this is why it's easier to justify that they're important to regular people, because domoic acid is a neurotoxin that kills people and animals. We don't know why some of them produce these. One possibility is that they function in biotic interactions. Maybe it's an anti-grazer. Maybe another idea, an old idea, is that it might help in competition for iron and copper because it also functions as a copper chelator. But Pseudonychia that produce this have been responsible for massive HAB events, harmful algal bloom events. This is for example, in 2015 and 2016, there was an enormous event that stretched from almost the border with Mexico all the way up to Canada, causing marine mammal deaths, lots of closures of shellfish fisheries all along the coast for quite a long time. Now, one of the problems with monitoring for Pseudonychia is as I said, they all look the same, and it's not possible to distinguish toxic versus non-toxic ones by the light microscope. Okay, so I was thinking this might be an interesting one to study, and I realized that there was a complete gap in knowledge in the Southeast Pacific when I got started. This is a review from 2018 in this map, and where there was all the node where they knew what species were found where, and basically they didn't know anything in front of here because there were some previous studies along the coast from very, very old ones that had used only electron microscopy, and nobody could tell really how to relate the species they'd seen to the modern definitions of species. Uh, People had used light microscopy and knew that they were an important part of the communities here, and they produced toxins here, but which ones they were wasn't known. So I thought, well, let's start with the basics. What species are there? We'll do the old-fashioned way, getting cultures, seeing who they are, describing new species if we have to, and testing whether they're toxic. And so that's what we did. So here's like this classic work, which the species definitions are based basically on these what they're called the ITS, the internal transcribed spacer 1 and spacer 2, between the genes for the ribosomal RNA 18S. And use the term genes a little bit because they're not exactly genes because they don't code for protein, but they're the ribosomal cistron has the section for the small subunit and the large subunit and the 5.8S here, and between those, these parts are transcribed, but then they're functionally they have to be cut out, and their folding is kind of specific or has to be conserved, but they're actually highly variable, so they can use to be distinguished between species. And you look at the folding patterns, and you look at if the bases change, and that's how people decide if two things are going to be considered the same species or not. Ideally, you have also do breeding studies. Now, you can do morphology, and sometimes the morphology also will different or overlap. And this is a case where we weren't sure enough to call it different species. Other people might have decided to call them a new species, but we found one that was offshore of Chile that looked very similar to one that had been previously described. It had some morphological differences and some molecular differences, and so that's just an example of what we were doing, and then making trees, and then finding, okay, well, when we're something so clearly very different from other stuff, then we said, okay, we'll have to describe a new species there. And previously there had been reported by older techniques 11 species in the Southeast Pacific, and now the number is 18 and supported by molecular evidence. And that number, with the limited sampling that we did, is actually equal to the total number of species reported for decades of sampling in the entire North Pacific. So it's very diverse down here. And curiously, I had just 2 samplings offshore near this oceanic island. In those 2 samplings, I got the same number of species and culture as I'd gotten for a much larger number of samplings along the coast. So this is just an old-fashioned culture-dependent method. The other thing that was curious was that the species along the shore, normally around half the species are toxic, and I found about a third of the species along the coast, consistent with what we expected, were toxic in culture, and there were species known to be toxic were the ones turning out to be toxic. No surprises. But offshore, even strains of species known to be toxic or considered to be toxic weren't toxic. So at least not in the culture conditions we tested. So that trend was one time hinted at before in an earlier study in the North Pacific that oceanic species tend to maybe express toxicity different. And if toxicity serves in some sort of interaction, either with competition or with like an allelopathic effect or an anti-grazer, nobody is really sure yet. That struck me as interesting. So we're going to keep investigating that. But I'm going to go back to a previous story we had, because something about this has been seen before. For a long time, people just assumed that phytoplankton, like other organisms, would always be in competition. And Siegel in 1998 did a simple physical analysis that phytoplankton in the open ocean really can't be competing directly for nutrients because the cells are too far apart. So the densities are too low. So they shouldn't be operating like that. John Dolan in 2007 published a paper looking at not at phytoplankton but at tintinid, the type of ciliate, and found their richness actually increases inversely with plankton biomass. So they're actually more diverse in these open ocean oligotrophic, ultra oligotrophic uh, areas, and the species rank abundance relationships were actually consistent with neutrality. That is, that they're not directly competing. And Behrenfeld in 2021 also discussed this idea that said that the open ocean landscape is probably much of the time is neutral with respect to competition, with except some times when something will be blooming or not. And I was paying attention to that because by accident I had discovered this strange change in the genome contents of, remember that species, Emiliania that's found everywhere? And I had been looking at it and found, by accident, found that the genomes of the strains that were coming from the open ocean tended to lose uh, the genes that are required for the flagellated haploid phase of the life cycle. So they couldn't produce this flagellated life cycle stage. And the only thing that I could relate that to was whether or not one had information about a high presence of a particular virus that attacks the other phase, the diploid phase of the life cycle. So it seems when it didn't seem like the losing these genes that we would find this shards of the genes, fragments of them. It wasn't being like they were getting any competitive advantage by having less DNA. They were just having non-functional genes. They're carrying around non-functional genes, and so that didn't look like the classical genome streamlining to be more efficient by getting rid of extra DNA. It looked like it was something that occurs when pressure is relaxing and allowing the things to have less selection to maintain. And so when there was less phytoplankton biomass and less of this specific virus attacking this one life cycle phase, that would be occurring. And that I think is something that also a lesson that might be learned is that there might be differences between coastal organisms and ones that live far away from the coast, and some of that might be based on how they use interactions with other organisms. Okay, so let's do some lessons. What are some lessons we're learning? As I said, I told now 3 stories that I haven't resolved yet, but we do have some lessons. So one is there are potential limits to phytoplankton adaptation. So one thinks of, oh, microorganisms, they divide so fast, they should be really good at adapting. But we saw in one situation that most phytoplankton can't colonize a sunlit oxycline for some reason, at least when it's over anoxic waters. Even though there's enough light there, there's tons of nutrients, there's CO2, all the things that phytoplankton need are there, and yet they're not colonizing it. So is there one possibility? Is there an unexpectedly high demand for oxygen? We usually think of phytoplankton as oxygen producers, but maybe they actually also need it. For example, for doing photorespiration or water-water cycles are always thought to be wasteful process, or used to be thought to be wasteful process, but perhaps they're necessary, and those processes have a high oxygen demand. Or maybe there's another factor. Maybe there's an inhibitor that's present in those waters that's being produced that causes it. But whatever the cause, we can show by a simple model that this is actually causing would cause a feedback with oxygen loss, and with, as you expand oxygen minimum zones, you might actually be decreasing phytoplankton by whatever is causing this. So another case of a limited adaptation is we saw that in the lab, the coccolithophores in the Chilean upwelling, they might actually be at the limits of their ability to adapt to ocean acidification already. Because in the lab, they really can't stand very well those conditions. They're not really good at it, but we still haven't resolved why we do have this phenotypic difference between these high pH, high CO2, low pH waters and the surrounding waters. We don't know yet why there's that, but it's not a simple explanation. One other case is that with coccolithophores, I pointed out that we often think of cells as regulating their homeostasis. That has to be more stable inside the cell than the surrounding environment. From a physiology class, it's usually where we're taught to think about organisms maintain the homeostasis inside. But in fact, phytoplankton, at least in the open ocean, are probably maintaining, they're going to be experiencing their metabolic fluctuations for processes like calcification, or if they're doing mixotrophy and they ingest an organism that they will then digest, or with the changes in the light throughout the day, they will have internal metabolic fluctuations that might actually be chemically larger in magnitude than the external medium normally, and they may be adapted to using the high stability of the external medium to help them maintain their own internal homeostasis. So then that could also push them to limits that we wouldn't expect for adaptation as the ocean chemistry changes. Okay, and finally, the last lesson is there's high phytoplankton diversity. We see neutral genomic losses and low toxicity seen in the Southeast Pacific gyre also suggest that things like resources competition and some of the other biological pressures might not always be the important drivers of phytoplankton ecology and evolution. So we go can't there might be situations when they're actually uh, like the open oligotrophic ocean, we have to maybe change our thinking of how those environments actually are. Maybe they're not always the in a highly competitive environment, so some of the evolution processes we see will be neutral. Okay, I'm gonna just show a picture of a bunch of people who are in my lab. Here's for instance, Victoria Solis. So we're doing the sampling with open ocean sampling. We use she lives on this island in Robinson Crusoe Island, and so we sometimes get to do open ocean sampling from a small vessel instead of trying to pay for a very big boat. Uh, Here's a bunch of people. The coccolithophore work was done by Francisco Diaz. These are people who helped me maintain the lab. Valentina has helped with getting a lot of the isolates of Pseudonychia and maintain them. Here, the Pseudonychia work is supported by the group in Italy in Stazione Zoologica Anton Dorn, and here they are uh, we're enjoying dinner together. Um, okay, with that, I'll thank you and I'll end and take questions. So hopefully you're all still there.
Okay, thank you